My name is Anirban Banerjee. I'm an investigator in the NICHD intramural program. And it's a real pleasure and an honor to introduce our speaker today, Chen, who comes to us from Rockefeller University, where she heads the laboratory of membrane biology and biophysics. Before I start sort of introducing Ju, I just thought I would share with you one. It's not a story. It's actually a fact that I heard about Ju back when she was an assistant professor starting a lab working on membrane protein structure function, and she was focused on this maltose transport system in E. coli. And back in those days, for those of you who are aficionados who would know that we were just learning how to purify even membrane proteins. And the norm, the wisdom would have been to screen a whole range of orthologs to figure out a well-behaved candidate and then go with that. So a more opportunistic approach. Ju, however, refused to do that because her point of view was that the most functional data at that stage for the maltose transport system was available for the E. coli protein. And she had a collaboration with Amy Davidson and said, I'm only going to solve the structure of the E. coli maltose transporter. And this would have seemed suicidal at that stage, but I think it also goes to show Ju's passionate love and interest for the science that she's interested in. She, of course, went on to solve the structure of the maltose transporter in many different states, obtained very deep mechanistic insights. And her lab has focused in general on a certain family of membrane transporters called ABC transporters. There are more than 2,000 of these. And then she has gone on to focus on very long-standing challenges in the ABC transporter field, such as P-glycoprotein, which is something our own Michael Gottsman has been involved with. She solved the structure of the human P-glycoprotein, which is involved in drug resistance of cancer cells, and most recently has solved the very long sought after molecular structure of the cystic fibrosis transmembrane conductance regulator, CFTR, which is the underlying sort of mechanistic connection between the disease, and has explained why actually an ABC transporter can function as a channel. Ju, of course, her list of achievements are very long. I'm just going to mention that she has been a Pew Scholar, is an elected member of the National Academy of Sciences, and also is an HHMI investigator. With that, I'll hand it over to Ju. Thank you very much for accepting our invitation. Look forward to your talk.
Thank you, Anirban, for this kind introduction. And it is my honor to give this talk in the presence of several founders and the colleagues of the ABC transporter field. Let me begin by telling you the birth of the ABC transporter field. In the summer of 1989, the scientific world celebrated a major breakthrough. Two press conferences were held on the same day, one in Toronto and one in Washington, DC. HHMI provided its private jet to make the schedule possible for the scientists involved. News about this discovery appeared all over the world, and all this happened 2 weeks before the scheduled publication date. Of course, this created a problem for the publisher. So the editors of Science ended up doing something very unusual. They lifted the normal embargo policy. So what's all this about? Here is what happened. The gene underlying a devastating disease called the cystic fibrosis was found. Through a collaborative effort between two labs, Li Ziyu's lab in University of Toronto and Francis Collins' lab at the University of Michigan at that time. So why is this such a big breakthrough in the field? Because cystic fibrosis is one of the most prevalent genetic disease in people from Northern Europe descendants. It is a recessive genetic disease. 1 in 30 Americans are carriers, which means they have one copy of the defective gene. Carriers are asymptomatic, but their children could be devastating. Knowing the gene means we can now diagnose cystic fibrosis by sequencing, identify the carriers, and it also means we can express the protein to study its structure and function. Cystic fibrosis triggers a cascade of problems. Mucus in the pancreas blocks cells from making digestive enzymes. So 90% of the patients have trouble digesting food and absorbing nutrients. Men with cystic fibrosis are infertile. And the most severe form of this disease occurs in the airway when mucus builds up in the lung, which leads to recurrent infections, chronic inflammation, and eventually lung failure. As you can see from this graph, on the x-axis are the time of the year and the y-axis are the mean survival age of patients. We can see that over the years, science had made a big difference in patients' life. Various treatments have been developed, which includes having a pancreatic enzyme supplements to help digest food, various ways to medicine to help clear the airway, and the antibiotics to treat and prevent infection, and also lung transplant to give patients new hope. So since the gene was identified here in 1989, we can possibly now through express the protein and high throughput screening, small molecules were now identified to directly act on the proteins and will modulate its function and expression. So in 2012, the first such a drug called the avicaftor, developed by Vertex, was approved by FDA. So, but even now, as of today, there's no cure for this disease. The average life span of patients is around 47 years. So what does the CFTR gene encode? This again related to another pioneer story related to NIH. So around the same time, CFTR gene was cloned. Michael Gottesman's lab and others were going after a different gene. This gene they named MDR is related to multidrug resistance of cancer cells. Here are just a few examples of publications came out of the Gottesman's lab around that time. So when this gene was finally cloned, it looks like just like a transporter. So it contains two homologous halves shown here, and each half containing a transmembrane domain and a nucleotide-binding domain, suggesting this could be an ATP-powered pump. And this structure is entirely consistent with earlier observation that multidrug resistance is energy dependent. And as Michael pointed out in his publication here, he noticed at that time, this protein, now they call it MDR1 or P-glycoprotein, is a homolog of other known pumps in bacteria, which also contains two copies of nucleotide-binding domain. So because of this homology, now they form this group of protein called ABC transporters, with ABC stands for ATP-binding cassette, which are the nucleotide-binding domains shown here. So when the CFTR gene was cloned, it came somewhat a surprise because it was known that cystic fibrosis was related to disruption of chloride permeation. Thus, it was assumed that the protein would be an ion channel and most likely a chloride channel. However, the gene, once it's being reviewed, it looks just like a glycoprotein. It also contains two homologous halves, each containing a transmembrane domain and a two ATP-binding cassette, except for a larger intracellular soluble domain, now we'll call the R domain. So therefore, because of this homology, it was thought CFTR might be a pump, and it could just pump out some regulatory molecules to regulate another chloride channel. That's why this gene was named cystic fibrosis transmembrane regulator. It turns out there is no other ion channels involved. CFTR itself is the ion channel. In fact, among the thousands of ABC transporters identified so far, CFTR is the only ion channel in the membrane, in the family. So here shown here is a recording of a patch of membrane. On the x-axis is the time and the y-axis is the current. And this patch contains only single CFTR molecule. So in this region of the record, the channel is silent, there is no current. And we add ATP, the channel is still silent. So that shows ATP itself cannot activate the channel. Treatment by protease K, protease kinase K, pKa, will phosphorylate the channel. And now in the presence of pKa and ATP, the channel flicks open so that we can observe current. pKa treatment, now we know, phosphorylate the intracellular domain, we now call the R domain. It will phosphorylate multiple sites in the R domain and allow the channel to respond to ATP. Once fully phosphorylated, we have a zoom-in view of this part, and you can see the channel now cycles between a closed state and an open state. We also know now, every time the channel opens, two ATP molecules bind, and then ATP get hydrolyzed, the channel closes. So every cycle, channel open and close, ATP molecule get hydrolyzed. So it's not surprising, pKa phosphorylation not only regulates the channel activity, it also regulates the ATPase activity of this molecule. Here are the ATPase turnover rate determined by purified CFTR protein as a function of ATP concentration. And you can see the dephosphorylated channel has very low ATPase activity. Phosphorylation by pKa activates not only the channel activity, but also ATP turnover rate. So in a way, CFTR is not only a strange ABC transporter because it kind of breaks down into an ion channel instead of moving the substrate uphill against its concentration gradient. A channel let chloride ion run down its concentration gradient. So it's a broken transporter. Also, it's a very strange ion channel because it is the only channel I know that consumes its own ligand. It uses ATP as a ligand. When ATP binds, the channel opens. But at the same time, it also hydrolyzes ATP, breaks it down into ADP, and the channel closes and it has to bind another ATP again to reopen the channel. So how does it work? To understand this, Let's first look at the structure of CFTR that corresponding to this part of the record. Well, the channel is not phosphorylated and in the absence of ATP. So the first structure of CFTR was determined by a postdoc Zhang in the lab, then from zebrafish, then Feng Yu, a graduate student, determined the human version of CFTR in the same conformation. The two structures look very similar. So I'm just presenting the human structure to show you the conformation. So it contains two halves of the molecule shown in green and blue here. This region is the transmembrane region, and here are the two nucleotide binding domains in the cytosol. And in between the two halves of the molecule, this red density represents the regulatory domain that needs to be phosphorylated by pKa. And in the structure, cryo-EM structure, we determined density for the rest of the molecules are very well defined, except for the regulatory domain, suggesting it's highly flexible. The ion conduction pathway, which is outlined here as a gray mesh, is located in between the two halves of the molecule. And in this particular conformation, is open to the intracellular side of the protein, the cell, and sealed off at the extracellular part. And this represents a closed channel. And the structure is consistent with mutagenesis studies summarized here. So CFTR contains a lot of arginine and lysine residues. Those that mutations indicate interfere with ion conduction are colored in purple and pink. The ones that has nothing to do with ion conduction are shown in yellow. As you can tell, the mutagenesis identified residues are all pointing to the ion conduction pathway. So another question we like to understand is the selectivity of CFTR. It is a general anion channel. It let many different monovalent negatively charged ions go through, but in the cell mostly means chloride ions. So now we're showing the structure as a surface representation, and the blue color means the regions that are charged positive, and the red color means negative charged. We tilted the molecules about 20 degrees so we can look down into the ion conduction pathway. And as you can tell from this zoom in view, is highly charged with positive charges. And those are because of the arginines and lysine residues pointing into the conduction pathway. So this structure supports CFTR pore is a funnel-shaped, sealed towards the extracellular part of the membrane, and it's lined up with positive charge residues. Therefore, it can attract negative charged ions through electrostatic interaction. So where are we in the CFTR gating cycle? So I just want to remind you, the structure of CFTR I just presented here represents a dephosphorylated channel in the absence of ATP. In the cartoon fashion, I can present it here. Those are the two transmembrane domains outlining the ion conduction pathway. And in this conformation, the two nucleotide binding domains are separate from each other. So next, let's look at the structure of CFTR once fully phosphorylated in the presence of ATP. So as a wild type CFTR in this condition will undergo cycles of opening and closure. So to stabilize a preferably open channel with ATP bond form, we use the same trick many colleagues used in the ABC transporter field. So we'll introduce the mutation into the nucleotide binding domain that will eliminate the ATP hydrolysis, but will not prevent ATP binding. So this mutation locked the channel mostly into the open conformation. So if you compare the two records here, with the wild type channel, the open probability is about 0.3, 0.4%. But the introduce of the mutation will increase the open probability to around 0.6 to 0.8. And the channel is mostly in the open configuration. So we determine the structure of the mutant channel in the presence of ATP after phosphorylation. And here's what it looks like. So this molecule now have a much elongated shape, and the two nucleotide binding domains come together, sandwich two ATP molecules at their interface, just like other ABC transporters. And the two half transmembrane domains also come close together to outline the open pore. So in this conformation, there is an open passage connecting the intracellular part to the outside. And this pore has a constriction site also close to the extracellular surface, but we do see an opening here right around where a lot of mutagenesis have identified key residues in ion conduction. So this, we believe this structure represents an open pore. So when we compare the two conformations, here is again the structure of CFTR in the dephosphorylated ATP free form. And once it's phosphorylated with ATP bind, the two halves of MBDs come together, and the two TMDs rotating relative to each other and opens the pore. So here's going back to the close the channel conformation. This time I like to zoom in to where the R domain is located. So in the absence of ATP and phosphorylation, the R domain is inserted in between the two halves of the molecule, and we observe a helix that's tucked in between helices, transmembrane helices 9 and 10 here in the TMD2 domains. So when ATP binds, the two MBDs come together, and this opening between the two helices no longer exists. These molecules move together, and now the R domain is relocated outside the intracellular opening and packs along MBD1. So by comparing those two conformations, we start to begin to understand how R domain regulates CFTR. The unphosphorylated R domain functions like an autoinhibitor tucked into between the two halves of the molecule, preventing MBD from coming together to form the closed conformation. And the phosphorylation will deposit a number of negative charge on this domain, perhaps makes it incompatible with this binding, with the binding side in between the two halves of the molecule. So the R domain will relocate outside the cleaved and allow the MBDs to come together. And only in this conformation, the channel opens, so ion conduction occurs. So next we ask, why is CFTR a channel, not a transporter? So to address that question, let's compare the structure of CFTR with a typical channel, for example, a typical pump, for example, PGP. So here the two structures of CFTR in the absence of ATP and in the presence of ATP. And the structures of PGP has been well studied in the field, including colleagues here at NIH, Casper Loher's lab and my lab. And collectively, we learned that P-glycoprotein, like many other ABC transporters, alternating between two basic conformations. One we call the inward-facing conformation, which is shown here. In the absence of substrate and ATP, the two nuclear binding domains are apart from each other, and the substrate binding site is located in the middle of the membrane and facing intracellularly. So we call this an inward-facing conformation. And the ATP binding will stabilize the MBDs in the dimerized conformation and reorientate the substrate binding site. Now it's facing outside the cell to release the substrate outside the cell. So in a cartoon fashion, this is the inward facing structure and this is the outward facing conformation. So as you can see, the structures of CFTR and the P-glycoprotein actually are not that different. The one layer of regulation that imposes on CFTR is the presence of R domain. Without phosphorylation, it tucks in between, so the two MBDs cannot come together. But once the R domain is phosphorylated and released from this auto-inhibitory position, ATP binding, just like in P-glycoprotein, allow the MBDs to come together. But what's different between the two structures is in this conformation. So now let's look at the P-glycoprotein in the outward-facing state or in the MBD dimerized state. Shown here is P-glycoprotein presented in the form of surface. So now you see we located, focus on this region, which we will call the intracellular gate. So in the ATP bond form, the two nuclear binding domains are together and this intracellular gate is closed. So we can see the helices forming the region are in close contact with each other. The very same region in PGP is different. In CFTR is different. So this is the structure of CFTR in the very same region. Now the corresponding helices are separate from each other, lined with arginines and lysine residues. And this opening creates a pathway for chloride ions to enter the channel. So in the MBD dimerized conformation, we can say the intracellular gate, which is normally observed in ABC transporters, is broken open. That's why the channel now the chloride ions can conduct between the two parts of the membrane. So to summarize this part, typical ABC transporters function through the alternating access model. The essence of the model is the substrate binding site can only open to one side of the membrane at a time. So when there's no ATP, it's facing inside. When there is ATP, it's facing outside. In this way, it can move substrate against its concentration gradient. And the principle for alternating access model is the substrate binding site should never open to both sides of the membrane at the same time, because if that happens, it will run down its concentration gradient instead of pumping uphill. And that's exactly what happened in CFTR. And as I just showed you, in the MBD dimerized state, the intracellular gate is cracked open and the chloride ions can go down its concentration gradient. So that's why we call CFTR is a broken transporter. And now you can be more specific. Its intracellular gate is broken in the presence of ATP. So next, we would like to just understand why mutations cause cystic fibrosis. It turns out the CFTR gene has many, many mutations. There are more than 1,000 mutations found in the gene. And most recent sequencing data indicate about 300 mutations will cause the disease, and depends on the nature of the mutation, the severeness of the symptom can vary. Among those 300 or 272 mutations, 53 of them are missense mutations, which means they change amino acid at 46 different positions. So people categorize those mutations into different types. Some mutations cause defect in synthesis, the gene will not make at all, and others will block either folding or trafficking intracellularly. And there's a third kind of mutations where just the protein can make the cell surface, but not very functional. So when we map those 46 positions into the structure, we roughly categorize them into several different groups. So one of them we show purple are the ones lining the ion conduction pathway, as shown here. So you imagine mutations or in those residues will either change the structure of the pore or the chemical environment of the pore. So it will affect the function of the channel. And the others are located, for example, close to the ATP binding site or the MBD dimerization site. So mutations in these regions will either block ATP binding or MBD dimerization and cause the channel to be not very functional. A third group of mutations could be folding mutants. One example is at the tip of transmembrane helix 1 and 3, two residues, glycine 85 and histidine 199, come to very close contact with each other, forming a key interaction between those two helices. Two mutations individually can cause cystic fibrosis, a glycine to E or histidine to Y. And either mutation is compatible with this packing. So we hypothesize those are folding mutants that will cause the channel not able to form this correct interaction. The most common mutations we see in patients are a deletion mutation of phenylalanine 508. And the phenylalanine 508 located at the interface between transmembrane helices and the MBD1. And it actually inserts into a hydrophobic pocket at the end of transmembrane helices 11. So without this residue, MBD1 can still fold, but this loop becomes much shorter and this key interaction is lost. So what happens is the protein can still be made, but this assembly of MBD into the transmembrane helices becoming very inefficient and the protein to be degraded by ER machine, protein quality control machines in the ER. So the majority of this mutant became degraded in the ER. So there are two kinds of small molecules have been identified to revert the effect of those mutations. For the one group of molecules we call the correctors, those are molecules that will help defective proteins such as delta 508 to fold and traffic to the cell surface. So those are folding chaperones. And the other group of small molecules we call potentiators. And those molecules act on CFTR that already made into the cell surface to increase its ion conduction. So as we show here, the potentiators can increase the channel activity even for the wild type CFTR. So this is a membrane patch, has thousands of CFTR channels in this case. So y-axis measures the current, and the treatment of pKa and ATP will increase the ion conduction, so you can suddenly measure the currents across the membrane. And now addition of a potentiator called the GLPG now increase the ion conduction by about twofold. So this group of potentiators not only work on wild type, but also works on a variety of different mutations. So G551D, for example, is one of the common mutation found in patients. In the absence of any potentiator, there's very little current, even in the presence of PK and ATP. But you can see the current increase as a function of concentration of the potentiator. We also measure the activity of potentiators in the membrane patch with a single channel. In this case, this membrane has two CFTR channels. You can see that this is the closed channel, and there's one channel open and the second channel open. So in the absence of potentiation, this channel has an open probability about 0.2. So addition of potentiator increase the channel openability probability. Now you can see more channels open more frequently. So we can quantify the effect. So this is the open probability of the wild type CFTR in the absence of potentiation around 0.2, and the addition of GLPG increase it to about 0.5. GLPG also increase the open probability even for the mutant. The same mutant will determine the ATP-bound conformation. So in the absence of potentiation, this mutant already have an open probability about 0.6, but the addition of GLPG increase it further to about 0.9. So now the channel stays open 90% of the time. So we determine the structure of CFTR in the presence of GLPG, and it binds to a very unusual place. So this is where the molecule binds in the transmembrane helices, where it breaks down into two halves in a hinge region inside the membrane. And this helices is quite unusual. We worked on many ABC transporters. We've never seen any other transporter has a broken transmembrane helices in this position. So when we compare the two structures of CFTR in the presence and absence of ATP, one is shown in yellow, the other one shown in blue here. On top of the global conformational change, where the NBDs come together and the two halves of TMDs come together, there's also a local conformational change happens exactly here around this hinge region. So there's a 55 degrees of local rotation upon ATP binding. So which means a channel opening not only requires the global conformational change of NBD dimerization, it also needs a local hinge rotation around for the half helices of transmembrane 8. And this is exactly where the potentiator binds. So the potentiator binds to the hinge region and presumably stabilize the channel, at least locally, in the open configuration conformation. So this is one drug. This is actually hasn't really made to the market yet. So this is a drug developed in a AbbVie, but also a drug that did make to the patients or developed by Vertex. It's called VX770. So it has a quite different chemical structure. But however, when we solved the structure of the Vertex drug, we were surprised to find it binds to exactly the same region, the hinge region on TM8. So when we compare the two structures, not only they bind to the same region, they actually interact with exactly the same set of residues in this area. So we carried out some competition assay. We can show that the presence of one drug will decrease the binding of the other drug, which add the confidence those two drugs compete for the same binding site. still, because for those of us working with membrane proteins, we know this binding site is inside the transmembrane region, and we often see densities can come from detergents, can come from lipids, can come from noise in the transmembrane region. So even though the density for both drugs seemed quite convincing because they are very different, they're absent from the potentiator-free structures, and each density corresponding to the chemistry structure of each compound, I was still not sure. So I talked to Fengyu, my student. I said, oh, Fengyu, you please make some mutations at the binding site, develop a binding assay, and see if these mutations changes the affinity of the drug. So Fengyu did that, not only mutate a few residues, she mutated every single residue that interacts with the drug. And then we find something very interesting. What we find out is when we change the residues that makes hydrogen bond with the compound, we have a dramatic effect. The binding is completely knocked out by this mutation. But when we change aromatic residues, for example, phenylalanine to aniline, we have some effect, and some various degrees of effects, but not as dramatic as hydrogen binding. And when we change other residues that make general hydrophobic interactions, we also have some effect and some has no effect. So this makes me think perhaps the different effects of residues have something to do with this binding site. It's a very shallow cave for the molecule. Half of the molecule is exposed to the membrane, and only a small part is buried by the protein. So perhaps in this highly hydrophobic environment, the interactions with hydrophilic residues, especially hydrogen binding, plays a more important role in stabilizing the drug in this particular environment. So we further validate the contribution of the two hydrogen binding residues, serine and tyrosine 304, using a functional assay. And again, you can see a single mutation will change the sensitivity of the drug in the membrane by about 30 to 50 fold. So with that, we can summarize what we've learned from our studies. Now we determined two conformations of CFTR. There's a lot more to learn about the gating cycle, and there's many missing steps. For example, ATP hydrolysis and the relationship between NBD dimerization and channel opening, the kinetics of those are all still need to be addressed. But what these two structures told us is how the R domains regulate CFTR, why phosphorylation is required to activate the channel. Most ligand-gated ion channels are regulated by the concentration of the ligand. When the ligand concentration is high, it will bind to the channel and it will gate the channel. And when the concentration changes, the channel activity changes. But a CFTR uses ATP as its ligand, and the intracellular concentration of ATP is quite constant. So its regulation actually indirectly through cyclic AMP. Cyclic AMP levels high, it will activate pKa, and the pKa will phosphorylate the R domain and will lift the R domain from its inhibitory position, allow the NBD to dimerize and the channel to open. Another thing we discussed is the structural feature that enabled CFTR to function as an ion channel. I see are twofold. One is the intracellular gate has a cracked opening, so which allows the ion to conduct to enter the pore in this NBD dimerized conformation. And the second feature is the break of transmembrane helix 8 into 2 halves and allow a hinge rotation of a local conformation that will further open the pore to the extracellular side. And this leads to the understanding of some mutations, not all mutations, why they cause cystic fibrosis. Some are defective in folding, some are incompatible for NBD dimerization or ATP binding. Lastly, we sought to address how at least the 2 potentiators, how they work. So those 2 potentiators bind to the site in the transmembrane region where local conformational change take place to allow channel opening. So with that, I'd like to thank people who are involved in the work. The structures I presented are determined by a postdoc, Zhang, and a graduate student, Feng Yu. They worked amazingly as a team, very productive. Now both of them are moved on. So now in the lab the project is being carried on by graduate student Jasper. I showed some of his single channel recordings here, gorgeous data, particularly from a graduate student. And also a postdoc, Carol, is in the lab study how CFTR folds and how the folding characters function. The PGP project, the inward facing structure were determined by former postdoc Min Song in collaboration with the technician Sarah. And the outward facing structure was determined by Yong Jin, a former postdoc in the lab, with some help with Michael Oldham. And I thank HHMI and the Cystic Fibrosis Foundation for funding. Thank you for your attention.
Thank you, Ju. That was just an amazing, amazing, amazing story. So unbelievable. It's just, it is also very depressing to watch as you know, how much can be done, you know, in less than a fraction of a lifetime. So it's a clear inspiration. Ju, if I may use my advantage as a host to ask a couple of questions. So the questions relate to sort of your correctors, if I may call it that, those 2 molecules that bind at that hinge site. Now, you also showed the most important mutation, the phenylalanine, that it's on a loop, and that binds at an interface. Now, where these small molecules are binding and where that phenylalanine inserts, those are 2 different places, right?
That's right, yeah.
So my question is that, I mean, of course, you already have drugs, but in principle, for this most deleterious mutation, the delta F508, because that looks like it's really a loop with a phenylalanine projected in. It's just perhaps a very naive question, but if you synthetically made a peptide, like a small peptide that is somehow the structure is stabilized, let's say with a disulfide, and you add that, do you think that in a delta 508, a peptide with that phenylalanine could actually insert in trans and correct the function? Do you know what I'm asking?
Yes, so you are saying is the defect of delta 508 seems to be the assembly of NBD1 into the transmembrane domain because a key interaction is missing. Can you have something here to glue them together to function?
Yeah, can you add a peptide in trans that actually has the phenylalanine? Can you add that, and can that actually recover the function, or it? Do you know that?
Yeah, it's an interesting idea. As far as I know, hasn't been done yet. Yeah, so no one has really tested. But I also want to point out is those 2 drugs showing here are potentiators. So okay, they are not the correctors. So those drugs work on molecules already folded, made to the cell surface, but they somehow cannot open or cannot open long enough to let enough chloride to go through. So to correct delta 508 defection, you need both. You need correctors that will help the delta 508 mutant to make to the cell surface, but you also need, on top of that, you need potentiators to make them function. So the patients are treated with a combination of both drugs.
I see. Okay, that's very interesting. So I just wanted to remind everyone that although the Zoom went down, that the CME code is 31306. I was asked to remind that, and for some strange reason the video cast went down for a while. But some people joined in the Zoom room itself. So does anyone have any questions?
So this is Michael, and first of all, let me welcome the dozen or so people who were able to join us on Zoom. And Ju, apparently the video cast went down sometime during your lecture, so the couple hundred people who were watching didn't hear the end of it, but it was recorded, and we will make sure that everybody knows how to access that recording. In looking at this figure which you're displaying now. Yeah. The two potentiators that you're looking at look suspiciously like they might be substrates for P-glycoprotein. That is, they're hydrophobic basic compounds. And I wondered if they are, and if they are, have you looked to see where they bind in PGP?
I haven't. This is a very good point. I should have thought about it, but I have not. If I have to bet, I think the Vertex probably already tested, because usually screening substrate for PGP is one of the first things people do in pharmaceutical companies. They try to avoid substrates of PGP because they would like to deliver the substrate into the membrane. But again, those molecules don't have to go very far into the membrane. This binding site is exactly in the center of the bilayer. So short answer is I don't know.
It would be interesting, and they might be nice probes to look at binding sites in P-glycoprotein.
Yeah.
Anyone else has questions? Or Michael, do you have any other questions?
Well, I see other people here who might well have questions because they're really experts in membrane.
Yeah, so if anyone else has questions, just please unmute yourself and go ahead.
So I have a question. Ju, really beautiful talk. I really enjoyed listening to it. So I'm thinking about the chloride permeation pathways in a number of different ion channels, like CLC chloride channels, like the TMEM16 cal spectral chloride channels. And in some of these other proteins, it's been quite ambiguous exactly where the permeation pathway is. And in part because it gets quite narrow, and at least in the TMEM16 proteins, it's very near the protein of an interface. And you showed very beautifully in the space filling diagram of that intracellular kind of fenestration, if you will, where ions can clearly enter and exit the internal pore. But I wonder, how big are the dimensions of the permeation pathway as you traverse through the membrane to reach the extracellular side? Is it quite large?
It narrows down to only about, it's smaller than a chloride can go through, basically. So it has to dilate a little bit further to open up. Yeah. But in our recent high resolution structures, we do see chloride binds in the mouth. So we do see the chloride binding sites.
Okay, so that was my second question, was because you showed that the pore is clearly very cationic, but is there actually ion binding sites where, some more sophisticated mechanism of ion selectivity may actually be taking place?
We believe they are. We're testing that idea. Yeah.
Thank you.
Anyone else has a question? Just unmute yourself and go ahead. Ju, I had a question about, I'm curious, this is perhaps a more less of a pointed question, and as much as you can share. I mean, now you've sort of achieved this landmark kind of conquest points. What do you think sort of lies ahead in the field of ABC transporter structure in general? Like, what are the next, sort of big goals? I mean, you're someone who can really kind of comment on this.
I can't speak for the field. I can only say what I am interested in.
Please.
Yeah. One thing, as I mentioned, I still think there's a lot of gaps in our knowledge in detail the mechanism in understand CFTR gating, PGP, the cycle, pumping cycle, PGP. That's one level. And also what I'm personally very interested in is the regulation. How nature regulate the activities of different ABC transporters, and how can we find the molecules? Like this case for CFTR, obviously, there are molecules identified to up or down regulate the activities of ABC transporters.
Got it. I'm curious, is the Delta 508 mutant stable enough that you can actually solve a structure of that?
We're working on that.
Okay, fantastic. Anyone else has any other questions? Well, if not, then Ju, you know that there's a virtual tea session which starts at, I think, 4:15. Now that we are done a little bit earlier, you can perhaps take a short break and then join. You have that Zoom link, which is a separate link.
Okay. Yeah.
And thank you very much once again for a splendid talk.
Thank you.
Yeah, let me add my thanks. It was a beautiful, elegant talk. I would say the best I've ever heard about ABC transporter. So, and I've heard a lot of ABC transporter talks. Really beautiful, beautiful work. Thank you, Ju, for sharing with us.
Thank you.